So welcome to this masterclass from Cambridge Leadership College. My name is Warwick. I'm one of the Z coaches at the Leadership College here in Cambridge. So in this masterclass, we're working on biology at AS level and looking at the Cambridge International Exams paper from winter 2014. This is paper 11. It's a multiple choice paper. And when the exam had been completed and the examiner's report was published, the examiner identified a number of questions that were particularly hard. There were 6 particularly hard questions. So we'll make sure we cover all of those in this masterclass. There were 10 questions that the examiner said that the students found relatively straightforward. So we'll probably skim through those. They're just ones that are fairly easy for you to look up for yourselves. And if we've got time, then we'll look at the questions that weren't straightforward and weren't the hardest, but were still worthy of attention if we have time in this masterclass. Okay, very good. So let's move on then to the first question. And I want to work on this question just briefly around this question of parallax errors. And it's given 3 experimental conditions here using a light microscope there, using measuring cylinder and measuring a biological specimen, and trying to understand which of them could have a parallax error. So just to illustrate what that is, I'm just going to look at this example here. If you had a biological specimen, they've given a shoot here. You had some kind of shoot that you had to measure, and there was a ruler, and you had graduations on the ruler there. Now, depending on how you look at the specimen through to the ruler, can give you one of these parallax errors. Now, if you have your eye here, then that's okay. You look straight down and you make a measurement there. But if you're off to one side a bit over here and you look down at the specimen, what's happening is you can see that measurement there. Or if your eye is over here, then as you look at the specimen, you're actually making that measurement there. And so these ones here and here are parallax errors. And the reason for the parallax error is because the position of the observer can move relative to the specimen here and the measuring apparatus, the ruler here. So this movement of the observer is the underlying cause for this parallax error. Now, so you can see there's a parallax error there when we tried to do a measurement of a specimen. And then the next one talks about a measuring cylinder. I'll just draw part of that, this one here. And as you have the meniscus there, and you have a scale. Now, depending on where you look, is going to affect the precision of the reading, or the accuracy of the reading. So if you look there, well, that's going to give you an accurate reading. If you're looking from above, then what you actually end up measuring is down there. And similarly, if you're looking from below, down here, then what happens is your measurement goes to that part of the meniscus there, and you're making an incorrect measurement. So, and that's why when you're taking a volume reading from a measuring cylinder, you've got to make sure that your eye is actually level with the meniscus. And the way that many people do that is actually to sort of crouch next to the laboratory bench to make sure that their eye is parallel to the bottom of the meniscus. But again, you can see this parallax error is caused by the possible movement in the position of the observer, the eye. And so parallax errors are possible for 2, using the measuring cylinder, and 3, measuring the length of the shoot. But in the eyepiece of a light microscope, then because the eye is essentially fixed, if you see what I mean, to the eyepiece, you locate your eye over the eyepiece, the opportunity for these parallax errors just isn't present. There's no chance of parallax error when you're using the light microscope. And so these parallax errors are going to occur in only 2 and 3, and the answer there is answer C. Now, I'm going to shoot forward a little bit because I really wanted to make sure that we cover the questions that the examiner has highlighted as the ones that the students found the hardest. We might just sprint back to some of these other questions a bit later on, but let's just make sure that we cover the questions that the examiner is saying were the hardest. And this one here was the first one that the examiner flagged up as the one of the 6 that the students found the very, very hardest to deal with in this paper. And I think possibly not because the underlying information is difficult to remember or the students don't know it, but the question seems to be presented in quite an awkward way. And very clearly you can see this is a question about primary, secondary, tertiary and quaternary structures in proteins and the kinds of bonds that are required in those different levels of structure. So let's just quickly revise those and make sure we've got an understanding of these different levels of protein structure and the type of bonds involved in those structures. Well, the first up, of course, is primary structure. And that's all about the peptide bonds, isn't it, that form between individual amino acids. All right. So if we look at those, then a peptide bond forms when you have a condensation reaction, and the carboxylic acid of one end of one amino acid has a reaction with the amine group of another amino acid, and you get this peptide bond. And this is the rest of the first amino acid over here, and this is the rest of the second amino acid over here. And so these peptide bonds are the kind of bonds that are required to form the primary structure in a protein. Now, secondary structure is when you get hydrogen bonding between the oxygen in this part of the amide link here and a hydrogen in an adjacent part of the amino acid. So you're getting a hydrogen bond from here to a hydrogen on a different peptide bond. And so that hydrogen bonding then generates a kind of a structure in the protein, either an alpha helix, or you can get a beta pleated sheet. And that's all because as these peptide bonds form, I'm not going to draw the whole amino acids, but you've got this repeating amide bond. I'm just kind of sketching it here. And what happens is that there can be a hydrogen bond between the oxygen there and some other hydrogen as part of an amide bond somewhere else in the protein chain. And that repeating pattern leads to either an alpha helix or a beta pleated sheet. Now, for the tertiary bonding, you've got this polymer of amino acids, and it can fold because there's some kind of interaction that causes the chain to fold. Now, those can be all different types of interactions. Some of them are strong covalent bonds, some of them are weaker intermolecular forces. And so the kind of bonds that can form that allow these protein chains to fold, well, you could have a thiol group from one amino acid and a thiol group from another amino acid, and those two can form a bond between the two sulfur atoms. I'll just draw that. And that bond between the two sulfur atoms is called a disulfide bond. So that's one way in which once the protein chain has been folded, that fold can be held in position because of this disulfide bond between the side chains of opposite amino acids here. Now, that's a strong covalent bond. Other kinds of bonding could be, again, hydrogen bond. And you can get another kind of bonding called van der Waals forces. And that's typically between two side chains, which are separated in the protein chain, but can attract each other through hydrophobic interactions. You have two side chains that are both hydrophobic, they're water-hating, but they're attracted to each other. And they can pull the protein chain into this folded shape through these hydrophobic interactions, or these van der Waals forces. And finally, you can get ionic interactions as well. So where you might have an amine side chain, which has become positively charged, and you might have a negatively charged carboxylate group. And that's electrostatic force of attraction between the positive charge on the amine and the negative charge on that part of the carboxylic acid is an ionic attraction. And so you've got 4 different ways in which the protein chain can be held within a fold. And that holding of this folded shape is the tertiary structure. Now, the quaternary structure means that instead of having looking just at a single protein chain, you're looking at 2 protein chains that are held together. And the same interactions that we had that were responsible for the folding of the tertiary structure, the ionic, the disulfide bond, the hydrogen bond, this van der Waals forces, this hydrophobic interaction, those interactions in the tertiary structure can fold the protein chain, but in the quaternary structure can bring 2 protein chains close together and hold them in position relative to each other. And the classic example of quaternary structure, I suppose, is hemoglobin, which has 4 protein chains, which all have primary, secondary, tertiary structure, but the 4 different chains are held together in the one large hemoglobin complex. So that's the different types of bonding that occur at these different levels of structure, and you've got to try and fit those into this grid here. And the only one that really fits is this one here, to say that the bonding that you can have between tertiary structure and quaternary structure can be, we saw it up there, didn't we, that the quaternary structure can be held together by ionic bonding. And that's true as well, that the tertiary structure is held together by ionic bonding as well. So this last row here is correct, because the quaternary structure and the tertiary structure are held together by, well, we have these 4 different types of bonding here, and they only give us 1 in the answer grid here. But ionic bonding, for sure, can hold together quaternary structure from separate protein chains. Ionic bonding as well can hold the folding of a peptide chain, a protein chain together through the kind of ionic interactions that we had up here. Okay, so the next one that the examiner pulled out for students as being particularly hard was question 27. So let's scan very quickly forward and try and pick that one up. Okay, and this is talking about the different phases of the cardiac cycle, the heartbeat. And it says that the sound that you can hear through a stethoscope is related to the valves in the heart. And so the question here then is, well, what's the first sound that you hear at a particular point in the cycle? And really the way to try and visualize this a little bit is to take a simplified drawing of the heart. So I'm going to just draw it like this. The heart has 4 chambers, doesn't it? And I'm leaving some gaps so the blood can come in and out of those chambers. And I should leave a gap here as well so that blood can flow from one chamber at the top to the chamber at the bottom. So the chambers at the top are the atria, and the chambers at the bottom, of course, are the ventricles. Now, and the ones here, they have rather thicker muscle around them, and particularly this one over here has the thickest muscle around it. And we're looking at it, so this is the right-hand side, and this is the left-hand side. Okay. Now, I'm just going to draw here a blue for deoxygenated blood. So this is blood coming in here because it's deoxygenated. The oxygen has been consumed through the process of respiration throughout the body, and the deoxygenated blood is coming into that right atrium. And later on it's going to get pumped out, isn't it? Because the deoxygenated blood needs to go off to the lungs to be oxygenated. And this is the coronary artery, isn't it? Now, the blood is going off to the lungs, it's going to get oxygenated there and return in here. And so this is now the, sorry, this is my mistake. This is the pulmonary artery, isn't it? I just remembered that as I started writing the pulmonary vein. So this is the pulmonary artery. And this is the pulmonary vein, the oxygenated blood coming from the lungs back into the heart. And then that oxygenated blood needs to be moved to the rest of the circulatory system, the head, and the rest of the body. And so that leaves, doesn't it, through the aorta. There. Okay, so then we've got the blood flow through the heart. And what happens, of course, is at the start of the cardiac cycle, there's going to be a compression of these atria to force blood from the atria down into the ventricles. And that compression of the atria is this atrial systole here. So that's the first step. Then once the blood has been moved into the ventricles, then there's going to be a ventricular systole. That's a compression of these ventricles, and the blood is going to get pumped around the body. And then once that contraction is finished, the heart relaxes. That's the diastole, and then at that point the blood flows into the heart again. And so then you can see, can't you, that when the blood has to move from the atria into the ventricles, at the end of that process, these valves here are going to close, because after this atrial systole has completed, when the ventricle systole happens and pushes the blood out, it's very, very important that these valves here have closed, so that when the contraction happens here in the ventricles, the blood can be pushed out. And so the first sound that you're going to hear after the atrial systole has finished is these valves here closing. And so those valves are the atrioventricular valves. And they're going to close after this systole has finished, so that when the compression happens here in the ventricles, then there's no blood flowing back to the atria where the blood has just come from. And so the first sound that you hear is this closing of the atrioventricular valves, which is the answer C. Okay, the next one that the examiner flagged up for students, as students finding the questions hard, was this section here through bronchus. And so what we're looking at here, here's the trachea, and then it's splitting, isn't it, into these primary bronchi, and then you get these splitting and splitting further, you get this bronchial tree, until eventually you get down to the bronchioles, which end in the air sac, don't they, the alveoli. And this is telling us that we're looking through a section of one of these a bronchus here, and we've got a very sort of close-up of the section. If we step back a bit, we'd see a whole tube. That's the air passage, isn't it? And the air passage is lined with these cilia here. And underneath those, you have, well, you've got the ciliated epithelium layer there. We have goblet cells that are secreting mucus. And underneath that, if I can just choose a different pen color, there's going to be a layer of smooth muscle. And underneath that, there's going to be a layer of cartilage. Now, as you go down this bronchial tree, the relative amounts of smooth muscle and cartilage change. So as you come down to smaller and smaller and smaller bronchi, down this bronchial tree, you get increasing amounts of smooth muscle and decreasing amounts of cartilage, until you get down to the bronchioles here, and then there's hardly, or there's no cartilage at all, and you've just got smooth muscle. So that's the structure. And the photomicrograph that we have here is just really zeroing in and looking at a little section there. And so what we've got, if you can just see them here, these are the cilia here. And then you can see in here, you've got these goblet cells that are secreting mucus. So right up here, this layer X then is called the ciliated epithelium. That's there. That's my red part in the diagram here. The next layer down, look carefully, you can see there's lots of fibers in there. So that is the smooth muscle. Just there. Okay. So we're just looking at this section through the wall of a bronchus. And so you've just got to talk about the functions of the tissues, all right? The functions of the tissues. And the function of the ciliated epithelium tissue is to move all the mucus that's being secreted. You can see these goblet cells here secreting mucus. That mucus has got to be, it's going to be trapping dust and some other pathogens as well. And that's got to be kept away from the alveoli, where the gas exchange is going to happen. And so that mucus that's being secreted by the goblet cells is being wafted by these cilii. It's being wafted up until eventually the it gets up to the buccal cavity, and that means it's going to be swallowed, and that's the way in which all the dust and pathogens that have been trapped in the mucus are removed from the lungs. So the function of the tissue X is to waft dust and dirt upwards. That's the function there. And this smooth muscle here, when it contracts, of course, it's going to make that airway smaller. It's kind of a circular muscle, isn't it, around the airway. And so its function there is going to make the airway smaller. It's going to constrict the airway. And so the answer the examiner is looking for here is the function of these tissues is, first of all, to the ciliated epithelium. It's going to be wafting the dust and other pathogens that have been trapped in the mucus. It's going to be wafting it upwards so that it can eventually be swallowed. And the smooth muscle layer there is used to constrict the airway. Now, the next one that the examiner flagged up for students finding particularly hard is this one here. There's a question about cholera and vaccination programs. Now, compared to some other vaccination programs, a vaccination program to eradicate cholera has not been successful. For example, compared to the vaccination program to eradicate smallpox, which was successful. So you've just got to understand in this question why, what is it about cholera that makes a vaccination program not effective? So we'll just talk about cholera for a moment. So cholera is bacterium, and first historical records of cholera actually were in the sort of medieval period in India. And although it was restricted initially to that geographical location, during the Industrial Revolution, with much more trade and much more travel between different continents, cholera was much more widely spread around the world and came into Europe during the 19th century. And cholera is spread when there's very, very poor sanitation standards. And so if there's water which is poorly sanitized, which has had contact with human feces, then the cholera bacteria will be present or can be present in that water. And if that water is drunk without any way of cleaning the water, then that's a way in which the cholera infection, the cholera bacteria can get into the body. Now, the body has a line of defense, of course, through the acid in the stomach. And actually, most cholera bacteria are killed by that first line of defense. And you can have something upwards of maybe 100 million of these cholera bacteria, and your stomach acid will do a great job in defending you against the bacteria. And it's only when you get particularly large numbers of cholera bacteria that the first line of defense in the stomach acid, is insufficient to overcome the large number of cholera bacteria that have been ingested. Now, once those bacteria have been ingested, then they move from the stomach into the small intestine, and in the small intestine, then the bacteria attach themselves to the lining of the intestine, or the epithelium membrane of the small intestine. And once attached, so that there's we'll just go through this because you'll understand how the vaccination works in a moment. So the attachment happens, and then subsequently a toxin is released. So you've got two steps there. First is attachment, second is the release of the toxin. And that toxin impacts the ion exchange in the cell, and the cell then loses a lot of water. And it's this process of loss of water which is the damaging part of the cholera infection, because eventually the patient becomes rapidly dehydrated. And it's that dehydration that evidenced through either vomiting or more typically diarrhea, and that massive dehydration eventually, in the cases of someone who dies from cholera, leads to a failure of the circulatory system, because there isn't sufficient fluid in the body to maintain adequate circulation. And so it's this dehydration effect that is the damaging aspect of this cholera infection on the human body. And the treatment, of course, is usually just to rehydrate the patient. So giving solutions of glucose and solutions of ions is sufficient to rehydrate the person and can lead to a recovery from the cholera infection. So that's a bit of background about the cholera, and the vaccines that have been developed target actually both stages of this infection. So the vaccine trigger antibodies that surround the bacteria and stop it attaching to the epithelium membrane, and the vaccines have antibodies that stop the toxin, the toxin as well. Now, the thing is that these vaccinations have been found to be never or not 100% effective, and also the vaccinations provide, they don't provide 100% protection. They provide protection initially, but then after maybe 2 or 3 years, the protection that they provide tails off. And so actually to have a successful vaccination program, you'd have to be continually vaccinating your population, because the vaccination is only, well, it's never giving 100% immunity, and in any case, that immunity is decaying quite steeply over a period of 2 or 3 years. So, a good answer to this question about why a vaccination program might fail is, well, it's not 100%. The vaccines aren't 100% effective, and anyway, the immunity that is acquired decreases quite rapidly over 2 or 3 years. But that's not really one of our options here. And the option that the examiner flags up is this one, to say that, well, we know that the cholera, once it's got through the first line of defense in the stomach acid, once it's got through that, or if it gets through that, then it attaches to the lining of the small intestine. And the examiner is just saying that because the cholera bacteria lives in the small intestine, the vaccines that are stimulated by antibodies, actually, generally, antibodies aren't that effective in the small intestine. So this is the one the examiner is flagging up. And really, these vaccination programs aren't the answer to cholera. When cholera first arrived in Europe in the 19th century, then the way of eradicating cholera, because Europe is now free of cholera, is mass public water hygiene programs. So making sure that drinking water is sanitized, and those kinds of processes of making sure that the water is sanitized, that's the way to eradicate cholera rather than vaccination. So some diseases such as smallpox, a mass vaccination program is effective in eradicating the disease. In the case of cholera, then the most effective way of eradicating the disease, and this has been shown in Europe, is proper sanitation of drinking water. And so, if to achieve eradication of cholera in other locations, then a much stronger strategy is sanitation and actually education, so that populations understand how they can treat water, for example, by boiling to make it safe to drink. That's a more effective strategy than a vaccination strategy. And we know from tests on cholera vaccines that they're not 100% effective, and they are only effective for 2 or 3 years. All right, so the last couple that the examiner flagged up for students were the last 2 questions on this paper, question 39 and question 40. And it's saying in a freshwater food chain, which is the least efficient energy transfer. So for a food chain, of course, you've got at the start of the food chain, you've got your producers, haven't you? These are these autotrophs. And typically we think of plants, don't we, there? And the plants are using photosynthesis to create glucose. So these producers are taking inorganic chemicals from the environment, particularly in the case of photosynthesis, carbon dioxide and water, and are using the process of photosynthesis to build glucose, which is then subsequently used by the plant to build more complex carbohydrates. So you have the producers. They're going to be consumed by primary consumers. This is the first trophic level, isn't it? And they're going to get consumed by secondary consumers. That's our next trophic level. And you can go all the way to the top of the food chain, and eventually you get your apex consumer. You might have, I don't know, 3, 4, 5 steps until you get to the, or 3, 4, 5 trophic levels until you get to the apex consumer. And as a rule of thumb, at each of these levels, only 10% of the energy is transferred. That's because, say, for this secondary consumer, which is preying on the primary consumer here, the primary consumer has got to use a lot of the material that it ingests for respiration, for all the activities that primary consumer needs to function as an organism. And so what happens is that 90% of the energy is used in the life functions of this consumer, and only 10% is passed on to the next trophic level, to the secondary consumer. So as a rule of thumb, there's about a 10% energy flow through these steps from one trophic level to another in a food chain. But the first step, where this producer is using sunlight to produce carbon dioxide, well, that is much lower than this 10%. So rough estimates are that something between 0.5 and maybe 2% of the energy that's available in light is actually efficiently or used, or energy is transferred in this photosynthesis step. Now, if we just look what's going on there, so if you have sunlight, and that, of course, covers the full spectrum. So it might include ultraviolet light, and then as you go across the spectrum, you've got violet and blue, go through green light, and then you've got orange and red, and then you've got infrared, haven't you, at this end of the spectrum here. And so all that light is in sunlight, but the pigments in the photosynthesis process, the pigments in the chloroplasts are only absorbing, there's a couple of peaks actually, they absorb the blue light there, and they absorb some of the red light there as well. And so this mid-range of the spectra isn't absorbed at all. It's either transmitted or reflected, and because the blue and the red are being absorbed by the pigments in photosynthesis, we see what's left over. We see the green. And so, this is the first step then, where because only some wavelengths and not all of the radiant energy coming from the sun is absorbed by the plants, then you could say that a lot of energy is lost in that first stage because only a couple of these wavelengths are being absorbed. And calculations suggest that as much as over almost 50% of the available energy is lost because the plant is only capturing the wavelengths in this blue spectrum range here and red there. Now, next, not all of this energy is going to be captured by the pigments in the chloroplast. Some of the light is going to fall on other parts of the plant, some of it's not going to be absorbed by the chloroplasts. And so another say 25% is lost because it's not absorbed by the chloroplast. It's absorbed by other organelles or other structures in the leaf and isn't used in the process of photosynthesis. Now, the process of photosynthesis with its light-dependent part and its light-independent part, the efficiency with which this radiant energy is used isn't that efficient either. And so you end up with more loss of radiant energy. And even when you've successfully used the radiant energy to build glucose, of course, the plant is going to consume some of the glucose that it's built during the process of photosynthesis through the process of respiration. And so at the end of this process, maybe only 0.5% to 2% of the radiant energy that was available has been converted to materials that can be passed on up the food chain. And that's because the glucose has then either been further polymerized to other polysaccharides, particularly, for example, starch would be a way in which it's a complex polysaccharide polymer, which is used as a storage of carbohydrate inside the plant. Now, just so you can see there that going through the different trophic levels here, your rule of thumb is that as you went up the trophic levels from the producer to a primary consumer, secondary, tertiary consumer, you've got a 10% energy transfer at each of these trophic levels. But the first one, the process of photosynthesis, has a very low energy transfer, much lower than these high trophic levels here. And just for comparison, just while we're here, solar panels are much better than photosynthesis. So solar panels are generally thought to be about 10% efficient at the moment. And probably as time goes by, these solar panels are going to become more and more efficient in capturing radiant energy and transferring it into useful energy. So for here, the answer for which is the least efficient is this first step here of photosynthesis. So these photosynthetic algae capturing sunlight, that energy transfer process through the process of photosynthesis has a low energy transfer value. And the examiner said that for most students thought that the energy transfer was least efficient at the top of the food chain. So I guess this answer here. It's true that as you get to the top of the food chain, there are larger numbers of the there's smaller numbers, aren't there, of the consumers of the higher trophic level. So that's where that thought is coming from. But in terms of the energy transfer, the efficiency of it, then the energy transfer is least efficient at the producer level, the photosynthetic level. Okay, the last one that the examiner flagged up for students was about the nitrogen cycle, and we'll dig into the question in a moment because it's quite a subtle question. But the examiner's comment was that most students had actually a very, very poor understanding of the nitrogen cycle. So let's just spend a little bit of time looking at that. So the nitrogen cycle is one of the cycles which involves biological aspects and kind of geochemical aspects as well. So nitrogen is present in the atmosphere, about 78%. And the nitrogen molecule is two nitrogen atoms triply bonded to each other. It's extremely strong bonding between these two nitrogen atoms. And so it's very, very difficult for organisms to use nitrogen directly from the atmosphere because of this strong bond between the two nitrogen atoms. But there are some organisms that can use nitrogen. And those kind of organisms, because they're using nitrogen directly from the atmosphere, are known as nitrogen fixing bacteria. And they can either be freely living in the soil, or they can be in a symbiotic relationship with plants as nodules in the root of leguminous plants. So these nitrogen fixing bacteria typically are in plant root nodules, or they're free in the soil. And the nitrogen fixing bacteria can capture this atmospheric nitrogen and turn it into ammonium ions. Let me try and draw that a bit more neatly. So NH4 plus is what I'm trying to write there. So that's the first step in the nitrogen cycle. The nitrogen in the atmosphere is fixed by these nitrogen, and the ammonium ion is produced, NH4 plus. Now, once that's available, then there are other bacteria called nitrifying bacteria. And they can convert the ammonium ion into nitrate ions, as NO2 minus, or further nitrifying bacteria can convert it one stage further to nitrate ions. So these nitrifying bacteria are creating these nitrate ions, and that is now a form in which can be absorbed by plant roots. So the nitrate ion is then ready to be absorbed by plant roots. And the plant needs this source of nitrogen, doesn't it? Because it needs to synthesize proteins and nucleic acids. Now, you remember from your structure of biological molecules, both proteins with the amine group and the nucleic acids with their nitrogenous bases both require a source of nitrogen. And that comes through the absorption of nitrates in the soil, which have come through the nitrogen-fixing bacteria to make the ammonium ion, and then the nitrifying bacteria, first to form the nitrite and then the nitrate. Now, in the soil as well, there are denitrifying bacteria. And they serve to take the nitrate ions and return that to nitrogen, atmospheric nitrogen. So we're almost complete with the nitrogen cycle. There's only one part that we're missing, is that as plants or animals die and decay, then there's another series of bacteria or, in fact, fungi involved in the decomposition process. So decaying or dead plant or animal parts are decomposed either by bacteria or fungi, and that decomposition process is going to release ammonia ions again, which can then be converted through to nitrate, so nitrate, nitrite, or nitrate. So that's the nitrogen cycle, and if you look back at these questions here, question answers here, we'll just pick up some of these. So question A, free-living nitrogen-fixing bacteria release organic nitrogen compounds into the soil. Well, that's true, isn't it? Because these nitrogen-fixing bacteria were either in the nodules of leguminous plants, or they were free-living, and they can release nitrogen compounds into the soil. We saw that with this ammonium ion that was being released into the soil. So that's true, because we're trying to find a statement that's not correct. Now, the nitrifying bacteria cause an increase in nitrite and nitrate ions. Now, that's true, isn't it? Because we had these nitrifying bacteria here, taking the ammonium ion and changing that into nitrite and nitrate. And once the nitrate is available, that's ready to be absorbed by the plants and is used to make proteins, because the amino group is necessary to build amino acids. They're going to later become part of a protein chain. So that's correct as well. Now, so we have in this section C, the nitrogen-fixing bacteria, the ones over here. They're fixing or removing nitrogen, atmospheric nitrogen. Now, the atmospheric nitrogen can be replaced by denitrifying bacteria. And so the denitrifying bacteria return nitrogen to the atmosphere. So that's correct. Now this question D is tricky, because it's true that these saprophytic and fungi, the saprophytic bacteria and fungi, they are decomposing organic nitrogen compounds. That's true, they are. So this question looks right, but then this is quite a subtle point. This decomposition, the question is saying, this decomposition is of decomposing organic nitrogen compounds that have been excreted and egested. Now, egested means that organisms are discharging undigested food as feces. That's the meaning of egested. Now, you can see that it's true that primary, secondary, tertiary consumers will egest organic nitrogen compounds in their feces. But it's not true that producers egest. Now, plants, of course, excrete oxygen. They, you know, there are things that plants excrete, but they don't excrete and egest. Egest is something which a primary, secondary, tertiary consumer is going to do. So it's not true that producers egest. And so for that reason, this is a false statement. Now you can see that's a pretty tricky question to get right. And it just means that it's really very, very important to read these questions with a very, very high degree of accuracy. Saprophytic bacteria and fungi are decomposing organic nitrogen compounds, but they're not decomposing organic nitrogen compounds that have been egested by producers, because that's not true. A producer doesn't egest feces. And so for that reason, that question is wrong. But when the examiner goes through this question, the examiner is saying that, well, even if you didn't spot that, you should have spotted that A was correct, B was correct, and C was correct. And so that only leaves D as the possible false answer here. Okay, so we've been through then this Biology AS level paper for Cambridge Leadership College. And if there's further questions that you have on that, because we've only had a chance really to go through the hardest questions the examiner flagged up, if there are further questions you've got about that, then the Z coaches from Cambridge Leadership College are going to be on the Slack channels, and we'll be delighted to take the questions that you've got through the Slack channels. So thank you for joining this masterclass from Cambridge Leadership College, and I wish you every success in your studies for AS level biology.